may I please take the pleasure of introducing Dr. Mohan Vemery to all of you. He is currently the Director R&D of Cell and Gene Therapy, Cell Biology Division at Thermo Fisher Scientific. Dr. Vemery has been with the company since 2006 in different R&D leadership roles and instrumental in stem cell-based product development for human pluripotent adult stem cell workflow and immune therapies in CGMP-regulated environment for applied research and cell therapy. He has served as faculty and staff at Children's Hospital of Philadelphia and worked on engineering stem cells for enhanced self-renewal, competitive engraftment, and immune reconstitution, utilizing novel gene transfer technology with hematopoietic stem cells. He designed stem cell-based assays and lead drug discovery using cell culture systems to treat Parkinson's disease in GLP settings at Thomas Jefferson University. Dr. Vemery currently teaches regenerative medicine at Johns Hopkins University, Baltimore, as adjunct professor, and he collaborates with different investigators in academia and industry to enable successful regenerative cell therapies in disease paradigms. And with this, may I please welcome Dr. Vemery as a speaker for today's webinar. Over to you.
Thank you, Nidhi, for such, and good evening to all of you, and good morning, Professor Redana. So before I open my talk, I have a few things to do as well. One is, I really appreciate the opportunity and thank Professor Redana for creating this FABA Academy. Particularly keeping in mind the students and the next generation kind of workforce that are required for this kind of emerging cellular therapies. As Manohar pointed out very nicely, Dr. Manohar, congratulations on the stem cell release in India and in the rest of the world. For those of you who do not know, I know Manohar was too modest in claiming what he has achieved. Thing is, if you look into mesenchymal stem cells all over the world, only there are 9 products in the entire world, and one of them is stem cell. There are other 2-3 products from Korea, and congratulations to Manohar for putting India on the global map for mesenchymal stem cell therapy, and it is heavily used. There are several clinical studies going on, but getting to this far and getting to see a product into the commercialization path is not an easy. He said 14 years or 15 years of journey, which is really difficult. The reason I'm pointing out that is, there are a lot of students attending this. It is not like, you think of an idea on Monday and come to a product on Friday, right? It takes a really, really long time to see. And this is one of the novel therapies because most of the therapies as of now are chemically treated with the small molecules, medicines, and all that. And in order for cells to be treated as drugs, a whole entire regulatory scenarios come into picture. You have to get through all the phases of clinical trials and get to that path of final phase 3 and phase 4 commercialization, which is not very easy. So thank you, Manohar, and congratulations for taking that very seriously and working through these hard times and all that. I really congratulate Cipla will take it up and distribute very nicely over the globe. So with that, I think, getting back to my talk, I kept it really simple as an overview, mainly keeping the students in mind as an younger generation in the mind to see where these opportunities are, why we should go after, and where does it make sense, something like that. A complete overview. And if any of you want more detailed kind of aspects or detailed discussions, I'll be happily available after the talk. And then Nidhi can provide my email to all of you. You can also reach out on email on any kind of subject that is related to cell therapy, cell and gene therapies. With that, Nidhi, will you allow me to share my screen?
Absolutely, sir. You have the rights.
Great.
Are you able to see the screen, Nidhi?
Yes.
Yes, sir.
Yeah, we can see more. Yeah.
So what I'm going to cover in today is, where are the cell therapies? Where are the cell therapies heading? And regenerative medicine is heading. What are the kind of new opportunities that are coming up at this level? Right. So if you consider cell therapy, there are a few other things that need to be considered. Most of them have originated from stem cells, and these stem cells can be differentiated into different cell types that constitute going into the organ developments. For example, nerve cells into the brain or cardiac cells into the heart. And when you get to this organization state of organ, it is not one stem cell. By the time there are multiple cells produced, differentiated, and organs have to work with the combination of multiple cell types orchestrated very nicely into an organ for development. And based on these ones, if you go back into cell therapy, now the cell therapy, it emerged, as Manohar pointed out, very early on with the adult stem cells, way back in 1960s by bone marrow transplantation. But as the time developed, there are new opportunities that have come up. And then adults are okay, but then at the time, for several disorders, availability of human cells becomes really difficult. From earlier, it used to be on the fetal sources, cadavers, and some of these ones. So new sources of cell therapy have come into picture. We'll talk some of those ones. And of late, there have been trials using gene therapy within the cells. So take the cells as a source and then modify the genes into one of those, any kind of monogenic diseases. And then by modifying the gene, putting back the cell in the same patient or in an allogenic manner, you could correct some of the diseases. That is also emerging very, very well. You will see that as well. And then the other aspect that is coming up is tissue engineering. So how can when the cells are like single cells, or when the cells are in the early stage, their behavior is different. They're expanding and some of these ones. But in order for the function, they have to be differentiated and has to be organized into a kind of tissue. And the tissue with the whole body has to be completely orchestrated and designed in such a way that the tissue works, the organ works in a way that is intended for in the human designing, right? So we'll cover these three aspects into the therapy. So this is what the whole kind of technology is. If you sum it up into a kind of a single statement, these are all called regenerative medicine technologies. Whether it is cell therapy, cell and gene therapy, or tissue engineering, they fall into this big bucket of regenerative medicine. So the main purpose of this regenerative medicine is a live cell therapy. Any cell that you are using is still live. It's not a dead cell. A dead cells won't work. So it is a live cell and it is a live modified cell. What is the whole idea of behind this cell therapy? Earlier I pointed out it was all medicines, capsules, tablets, surgery, and all this. There are other ways of doing this kind of treatment for certain diseases, and this is mainly targeted at even protecting some of the systems within the human body and repair some of the systems, like what Manohar has done in case of the limb ischemia kind of scenarios, repair it. And then in certain diseases like regeneration also. So why is regeneration? Regeneration is, for example, Parkinson's disease. The cells degenerate, right? So is there possibility to regenerate some of those very early dying neurons, right? So that is a kind of regeneration. So mainly a protection, providing a kind of repair process, providing a kind of regeneration, mainly to improve the quality of life in individuals. So this is the kind of NIH kind of definition that I'm not going through word by word, but mainly it includes cell therapies, tissue engineering products, and the purpose is to either protect, repair, and replace and regenerate organs, not only at the cellular level, even at the organ level, and manipulating through their genes or metabolic processes, some of those kind of mechanistic aspects on that. So the regenerative medicine has come into the mainly for the practice for the past 10 years is that it will alter the course of medicine. I know there are some doctors in this talk attending, it is definitely going to take a revolutionary approach. 10 years back, Manohar, in fact, organized a conference in Bangalore where the discussion mostly was centered around is stem cells a hype or a hope. It is not the question of hype anymore. We are hitting the ground with hard realities and successful products like the Stebucil that Manohar pointed out. So we are addressing a lot of disorders using this kind of cell therapies and regenerative medicines. So how it is evolving? So if you take cell therapy is mainly, and I mentioned that you need to have really viable cells or purified cells from a source. Like what Manohar said is he purified mesenchymal stem cells from pooled donors, and it is being used in an allogenic manner. Allogenic, for some of you very early on in this study, is if cells are taken from one person and those cells are modified, expanded, or manipulated and given back to the same person, these are called autologous therapies. Most doctors have direct authority to do this. On the other hand, if you identify a healthy donor, get the cells, manufacture it in a manufacturing facility like a GMP, what Manohar has done, and then give those cells for different indications for different patients. That's what is the allogenic therapy. So cell therapy comprises autologous and allogenic therapies. Keep this in mind. At any point of time, we need those kind of autologous and allogenic therapies. Allogenic therapies won't work if your own cells can go against to it. So therefore, there are a lot of techniques that need to overcome those kind of immune suppression and various approaches that you need to take if one is heading towards allogenic therapies. On the other hand, the gene therapy I mentioned. So within the gene therapy space, what happens is you introduce genes. There are already genes within the human body, and we know where the disease is. So go back to that site of human genes and then either replace it, or if it is very simple, modify it in vivo and then deliver the correct gene, and by either both to durability of treating for prevention. That is a durability, meaning that if you do one-time correction, that should be systemic and should be extending all over the lifespan of the individual. That's the durable treatment. He doesn't have to go back to hospital for repeated injections or repeated treatment of this kind of therapies. Those are possible with gene therapies. And either preventing and potentially curing diseases. Several times, mostly it is being used in several types of cancer. And in no time, it will also be done for autoimmune disorders as well, purely. And in all this, the gene therapy becomes very important for viral diseases as well as inherited disorders. So for example, viral disease, COVID-19 is very clearly imprinting all over the world. How do you treat using a gene therapy if we don't know the genes that infest and manipulate in the human body? If that can be prevented, that can be controlled. It is not a quick and easy answer, but then that is the way people have to go on it. So the other aspect is the tissue engineering that I mentioned. Generate tissues, transplantable tissues as such. So that is one of the tissue engineering. That is also an emerging area. Which is very early stages of tissue engineering, and that is one of the kind of things. So in order for gene therapy to do, one needs to understand genome editing tools. So these genome editing tools, how do you manipulate the genes precisely, right? So if it is one nucleotide change, can you change that? Or if it is a segment of nucleotides within a gene, can you replace it? So very early on, there have been techniques like zinc finger nucleases, and then TALENs, and then the CRISPR-Cas9 technology, which came up about 10 years ago. So this year, incidentally, 2020, just a week back, Nobel Prize was awarded to Jennifer Doudna and Emmanuel Carpentier for developing this CRISPR-Cas9 technologies. So this CRISPR-Cas9 technology, the real advantage with that is you can very precisely cut like a scissor, cutting a cloth. We can exactly cut in the place and then replace that gene or modify that gene without having any off-target effects. What I mean by off-target effects is just like any chemical drugs have a side effect, gene therapies also will have side effects. That is the off-target effect. You intend for something, something else happens. Those kind of things are completely covered, completely cleared up using this kind of CRISPR-Cas9 technologies that was originally from bacterial development, from phage kind of development that happened 10 years ago. And I'm very glad to see that this effort has been realized, Nobel Prize, just last week. Just to tell you, the reason I tell you is it took 10 years to get the Nobel Prize, which is really fast in that case. And these technologies are really, really emerging in kind of, at one point of time when Professor Redana and I were students, PCR is a big thing. Now these kind of technologies have to come up really well for the future. For the next 10 to 15 years, it's going to have a really big field in that. So why are we focusing on this, right? So why the students have to think about it? So if you look at the cell therapy sector, I'm constantly going to use cell therapy and regenerative medicine as an alternative kind of thing, but they mean the same to me, in my opinion, right? So if you look at the sector growth in the whole world, there have been more than 1,000 trials, right? And this is the global distribution of clinical trials at different stages, phase 1, phase 2, phase 3, and commercialization. So it is globally distributed. And that's why I was saying, if you look the Indian map, that's why I was saying Therapeutics is on the, put the India on the global map. And if you sum it up, all the different number of clinical trials, we'll go back to these numbers again later. If you sum it up, what is the whole revenue that is present in the cell and gene therapy? It is about $10.7 billion, right? So, and it is increasing year by year. For example, the 2020 in Q2 versus Q1, you could see almost is doubled up. And if you split it down again, gene and gene modified cells, about $7.9 billion. Cell and therapy, these are all combination. There is an overlap in these numbers, but keep that in mind. The cell therapy alone is $7.5 billion, and tissue engineering anywhere about $84 million. So there's a lot of money invested on the side of healthcare, right? So it is, and it is more than 10 to 15 years investment that's happening, increasing year by year, which directly tells the students that this is a very highly emerging opportunity of growth of cell therapy that's happening currently in the world, in all parts of the globe. So if you take these 1,000 trials and look it up, phase 1 is about 309, close to 400, let's say, and phase 2 is roughly about 580, and phase 3 is about 97. So what it means generally is, most of the time, only 1/3 of the studies come to a phase 3 level, and from there it further gets down onto really commercializable technologies. So many of them fizzle out, either because of safety, or because of efficacy, or because of other stringent regulations in countries like India, Korea, in US, and all that, right? So there's but the pointer here is, even though 1,000 trials are going on, almost 1/10 of it, like 97 trials, are getting to realistic manner in terms of commercialization and standard, and can be used as a standard of care that doctors can prescribe for different diseases, for devastating diseases. So if you look into the clinical trials in terms of this, we mentioned already there are almost different disturbance. But most importantly, what I want to bring about for students here is, of the 1,000 trials, almost 660 are in oncology area, right? So if you look into this immuno-oncology area, these are 665 trials, meaning that there's a kind of skewing. A lot of trials are happening in this kind of oncology area, and that is an area that is being concentrated. And followed by that, you have musculoskeletal, some other diseases like chronic limb ischemia, critical limb ischemia that Manohar pointed out, and endocrine and metabolic genetic disorders like diseases like diabetes disease, and central nervous system disorders for neural like kind of disorders are heavily focused in this clinical trial towards commercialization and the cell therapy approaches. So coming back to sum it up, there are a lot of trials going on all over the globe. The area is really emerging. And this is a kind of summary slide that I want to put in briefly here. The cell therapy consists of primary cell-based therapies, meaning that from bone marrow, the cells from bone marrow, adipose tissue, and some other differentiated tissue, from human tissue, cells can be extracted and further could be used for cell therapies. The other kind of therapy that is also happening is in terms of immune cells, particularly T cells, natural killer cells, dendritic cells, macrophages. They are all currently being used as an immune therapy. And combining these two is you have cell and gene therapy. So if any of these cells are used, irrespective of the cell source, if the genes are modified and if the genes are manipulated, then you call that as cell and gene therapy because the cell comprises the cell, it comprises a gene as well. And there are therapies like stem cells as such, right? So bone marrow derived stem cells, mesenchymal stem cells, human embryonic stem cells, and immune pluripotent cells, not immune pluripotent, induced pluripotent cells, which can be used in autologous and allogenic modalities. We'll talk of autologous and allogenic a little bit here. Autologous in the sense, a patient goes into the hospital, the doctor identifies what is the disease, and he in a kind of team, institutional guidance, believes that his cells can be taken out and modified and could be put back into the same patient. That is autologous therapy. And if you look into the logistics, the patient has to come to hospital, the doctor has to identify the disease and the right type of cell that he has to be manipulated, and then isolate those cells, culture those cells, manipulate those cells, expand those critical cells, and put back into the same patient, which means you need a clinical facility, you need a site for treatment, you need the patient has to come back. So there is a kind of time, there's a kind of latency associated. There's a kind of time lag. The cells will be taken out and the patient will be sent back home and asked to come back again after 15 days or 16 days or whatever is the time frame. And then the cells will be administered. And the doctor at that point do not know whether the cell dose is sufficient. If it is not sufficient, they have to come back again, right? So those kind of therapies are autologous. The reason I elaborate that one is you compare that to allogenic. In the allogenic product, like what Stempeutics has done, if the allogenic product is readily available on the shelf, which has gone through the process of clinical testing, evaluation, and commercialization, and you have a vial ready to be injected for a specific disorder, which means the patient doesn't have to wait. So there is the lag of time is cut off completely, and the doctor can decide whether whether the cells are in scope or out of scope to treat a particular disease for a particular person. The real advantage here is you have a readily available cell therapy that could be administered to patients. That is the real advantage in allogenic therapies. On the commercialization aspect, because you make a large scale of cells, you need to go through tough master bank creation, working bank creation, and evaluating it, creating the efficacy, and therefore you have cost of goods completely gets reduced when it is administered over a population, right? So that is on the commercialization end of the mind on that kind of autologous and allogenic therapy. For patient, the treatment is available immediately. For the company that is doing, for the pharmaceutical or firm that is doing, it has reduced costs, so it can commercialize very well and it can survive well. So with that, I will get into the other kind of technology that has popping up very nicely. This is a Nobel Prize work of Shinya Yamanaka in 2012. And these are called induced pluripotent stem cells. And these are very similar to human embryonic stem cells. Earlier in 1998, people identified human embryonic stem cells that could be taken out and could be differentiated into multiple cell types like neurons, kidney cells, whatever cells you may want to. But there were issues. The issues being, these are embryonic derived and fetal derived. Therefore, there are ethical issues. Can you touch the embryo? Can you take the embryo at what stage? So that became a realistic problem. Availability became a real problem on that case. What Shinya Yamanaka did in 2012, over the past years of 5 years ahead of it, is that he takes the cells from a patient like this, and using 4 transcription factors called c-MYC, OCT4, KLF4, and SOX2. So you can take any differentiated cell, like skin cell, for example, or a blood cell, and if you treat them with these transcription factors for a little bit of time, or 15 days or 20 days, then automatically these iPSC cells are forming. So the induced pluripotent stem cells I'm calling as iPSC, which are, I would say 99% are similar to human embryonic stem cells. Because of this one, you can take cells from differentiated cells from a patient from any tissue and can send it back by turning and making like a U-turn, sending into an embryonic stage, and we call them as pluripotent stem cells. Pluripotent means it can generate any kind of organ in the body. Plural, potent is potential, pluri means many. It can generate many, many different organs in the human body, different cell types that are needed in the human organ. So the very first advantage that came up with that, you can take the cells from the patients, you can repair those cells, differentiate them, and put them back in the same patient. That's one of the treatment. That's one treatment. The second advantage that came up is from the same cells that are identified from iPSC, differentiate the cell type, do all the in vitro testing with all the chemical compounds that you need or any disease-specific drugs and all that, and see whether that works for the same patient. That means without going to the patient, you can run the clinical trial in a culture flask, in a, let's say in a 96 well flat, in a 96 well plate, you can run those kind of cultures. So this work was awarded in 2012 Nobel Prize. So one important factor here is the iPSC cells themselves cannot be injected into a patient because they will form tumors, right? So therefore you have to differentiate the desired cell type that you want, and the desired cell type could be after differentiation could be transferred into the patient for treatment. So basically any kind of cell which can form the three basic germ layers like ectoderm, mesoderm, and endoderm. I bring up this because when human development takes place, it is zygote first. After some stage, like by 7th day or 10th day around, it forms three germ layers. And these are the three germ layers. Ectoderm, which means it gives, the ectoderm gives rise to all the surface organs that you see, like skin, including brain and all those. So mesoderm, it clearly gives rise to organs like cardiac and the blood and all that. And endoderm gives rise to organs like lungs and pancreas and some of those kind of the really important organs and all. But summing up, any organ in the human body comes from these three basic germ layers. And iPSCs can be differentiated into any of the cell types having that. So that is the main reason why they call it as pluripotent stem cells. So now these are currently being used not only in disease modeling and drug discovery, very close to humans without going into monkey and mice and all that. You can directly do the in vitro test on the human cells, on the human population that you are intended. And these cells can also be transferred into cell therapy models, which are currently emerging for diseases like age-related macular degeneration, diabetic retinopathy. These cells are currently being used on those studies. That's one of the things. So the other ones are adult stem cells. Adult stem cells meaning there are many adult stem cells. This is one mesenchymal stem cells. These are also called mesenchymal stromal cells. This is one of the very early cell therapies that have come up. You have a very clear, from any kind of tissue, they could be isolated like bone marrow, adipose, umbilical cord, placenta. Some of these sources they can be isolated and they can be expanded and could be used for different diseases. There's a clear marcation, like, what could so these cells could be differentiated into mesodermal derivatives like chondrocytes for cartilage diseases, osteocytes for bone disorders, and adipose disorders for fat-related diseases, and even some of those kind of like tendons, like tenocytes or muscle cells, they could be differentiated into all kind of mesodermal cells. And there is also a new idea now. Any iPSC that is derived from this mesenchymal stromal cells is also highly useful in terms of stem cell therapies on that. So one of the real advantages that is seen, these cells are currently being used for aged elderly diseases, mainly the knee issues, the limb issues, those kind of things, and bone-related disorders are heavily being exploited with this kind of mesenchymal stromal cells to repair and even prevent the knee replacement and cartilage replacement. And apart from, there was a company called Tyzonex, which is taken over by Takeda. They are working with chondrocytes very heavily for tissue repair, for cartilage repair. That really happens most of the age level 50 and above. You see knee issues and cartilage issues. Those can be corrected using this kind of cell types. So the other therapies that are happening fully is anti-tumor immunotherapies. So if this is a cancer cell, there are many ways to tease it out and many ways to approach killing the cancer cell. One of the very earliest approaches was the hematopoietic stem cell gene therapy. So hematopoietic stem cells are taken out from a patient and can be gene modified through a viral delivery. We'll talk of viral delivery in the next slide. And after modifying the genes, they can be put back in this patient. The original hematopoietic transplant studies took place somewhere in 1960s, and it is evolving very highly, both in terms of autologous and in kind of allogenic therapies. I mentioned to you already what is autologous and allogenic. I'm not going to go through it. So cancer can be treated or is currently being treated by 4 different cell types mainly. One is the T cells. We'll talk of one cell type therapy called CAR T therapy in the next slide. The other one is the hematopoietic stem cells that we talked about. And the other one is the natural killer cell. This is the CAR T therapy. And then the other one is the natural killer cells. Now the human body has natural killer cells. These are CD3 negative, CD56 positive kind of cells. This is completely opposite of T cells, the natural killer cells. And the other therapy that is also heading very nicely is dendritic cell-based immune therapy. So if someone has to create a vaccine, dendritic cell is important. When a virus or some chemical or some kind of poisoning takes place within the human body, then the first cell to respond is the dendritic cell that identifies it as a foreign invader and tries to attack and kill it and take that antigen and present it to T cells. Then the T cells build up the immunity to fight against that viral infection or whatever may be the intruder causing a disease on that. So these therapies are coming up, and these therapies are currently being used and in different stages again. So if you take the hematopoietic stem cells, either from the bone marrow or from the peripheral blood, the cells are isolated. The stem, mainly here, the CD34 called stem cells, CD34 cells, they're isolated. After isolating, they go through 2 different pathways. One is the gene editing transfection. It could be done by transfection agents. There are several agents that could allow, the transfection is nothing but putting a gene inside. It is like an adjuvant when you create an antibody. So this by transfection, you can get in the gene into that, and then you correct the cell. After correcting the cell, you can put it back in the same patient. Similarly, the other way is you can take there are kind of viruses like AAV and lentiviral vectors and adeno-associated virus vectors, which are human compatible. You disable the virus first and pack that gene into this virus as a vector and put that vector in the stem cell and put back in the patient to correct it. Right? So these are hematopoietic stem cells. What are the kind of diseases that are being addressed on this one? For example, sickle cell disease. You all know what is a sickle cell disease. The red blood cell becomes like a sickle shape, and then there is a lot of issues associated with respiration and a few other aspects with the sickle cell, and then it causes anemic as well. So those kind of diseases where there is a single gene that is affected is being worked out using hematopoietic stem cell based gene therapies. Thalassemia, beta thalassemia, is also a globin gene that is getting affected in this case. That is being addressed using hematopoietic stem cells. HIV, we know the CD4 cells, CD4 helper cells are the ones that are impacted in the HIV. So people are working on taking hematopoietic stem cells, going up, making them into CD4 cells, transfer those cells to HIV patients to come up. So the gene modified cells, one of the advantages here is that once you modify the genes in the HSC and put back in the patient, they get into bone marrow. Once they are into bone marrow, once they engraft, that is called engraftment. Once they are engraftment and improve the disease-free survival and quality of life of patients is for the rest of the lifetime in these patients. So the longevity of the patients is highly advantageous for getting this engrafted studies in the bone marrow, which means you are systemically correcting it, not a topical. You are systemically correcting the whole system within the human body. So in order for this long-term protection against disease, the HSCs that you have either engineered or you have isolated, they need to be all through the lifetime. They should be retaining the engraftment, proliferation, and differentiation. All of you know the red blood cell, for example, stays only for 3 months in the human body. What you have the cell today, the RBC, is not there after 3 months. It's died. It's dead and gone. So that has to be replaced. So that replacement has to happen with a gene modified and corrected cell that is present in the bone marrow, that is engrafted into the bone marrow when you did this treatment. So that is the real advantage here for long-term recovery, survival is the main advantage using this kind of immunotherapies or hematopoietic stem cell based therapies. The other one that is coming up very recently, a couple of years back, FDA approved this as a rapid therapy for certain patients. So the disease here is a acute lymphoblastic leukemia, which is a blood cancer disorder, So one of the real advantages here is that the real patients that are chosen for this is, as I mentioned, cancer can be treated if it is a visible tumor that could be surgically taken out, or they will go through chemo, or they will go through radiation. These are the conventional treatments that are present. Once the cancer becomes resistant to all these modes of operation, like blood cancer, there's no way to do the surgery for a blood cancer, right? It has to go through chemo or radiation. And if these treatments fail, the tumor becomes radio-resistant, chemo-resistant, and surgically not available. At this point of time, what the doctors would say is hospice, meaning prescribing death and asking the patient to stay home, right? There is no way of other treatment. What has been done in this case is, the T cells are the immune, they are like a police in the whole system in the body. They know where the tumor is, but their cells are not enough. The number is not enough. There is not enough force for the cells to cover. So therefore, what one of the researchers at University of Pennsylvania called Carl June has done is take those cells out, they isolate the T cells from this patient, engineer them, put a tumor-specific antigen on them using the CRISPR-Cas9 technology that I mentioned very early on, for which Nobel Prize was awarded. So using this technology, he puts in a CD19 for this acute lymphoblastic leukemia antibody on the T cells and expands them in 15 days and puts them back in the patient. So what exactly is happening is the CAR T cells are developed, and the T cells are isolated, engineered, and expanded on these T cells, and these CAR T cells are put back in the patient to get the relief in these patients. So this was started about 8 or 9 years ago, and the first patient is still surviving very well on this technology. So CAR T therapies are, so they tell you the impact of this is that this idea was brought by a $30 million investment into University of Pennsylvania. This study has come from University of Pennsylvania from Carl June lab, and Novartis company paid $37 million for this invention, and they take it further into clinical trials and all that, which is now approved as a rapid drug for this treatment. So, and just starting in 2018 and 2019, just based on this technology with small variations to different indications and all that, there are almost about 67 companies now working in this area of CAR T therapy. You could see within 1 year how this kind of invention could lead into translational therapies on that. So this is what is done. There's a cancer cell, there's a clear target identification using this chimeric, so the CAR stands for chimeric antigen receptor engineered T cell. So this T cell can bind and kill the cancer cell. So you increase the population of these T cells, right? This is the process by which these CAR T cells are manufactured. I'm not going to go into detail, simple. There's an apheresis step, there's a T cell selection, T cell activation, and a gene transfer. After that, you have a T cell expansion, and ultimately the patient is given to this. So just to tell my role in all this is, I work with all of these processes. Virtually every process here is being supplied by reagents or tools that we make at Thermo Fisher for this kind of therapies, right? So I mentioned about ex vivo and in vivo gene therapy with the, in order for the cell, you create a cell that is engineered, right? So that is a big task. So you have clearly identified that and you have expanded it. So after expansion, that is not enough in these cell therapies. You need a delivery. So for this delivery, you need viruses are being used. By dismantling them, by inactivating them, those viruses are currently being used into the cells, either as a direct delivery. So you can take a virus, inactivate it, put the gene in it for monogenic diseases like sickle cell disease, and you can put it into the patient. On the other hand, the hematopoietic cells or T cells or CD4 cells could be inserted with any kind of gene of interest, and it could be put back, cultured in the culture dish, and then put back into the patient on this kind of. The gene delivery and cell-based delivery using transfection agents is also very, very important. The viral, which means the viruses, we all know they infect. Taking those viruses and converting them into a clinically usable product is another area of research that's heavily impinging and growing in the kind of cell therapy and regenerative therapies. So I come to the last part. I know there are only $84 million investment in this particular area of tissue engineering. I don't know whether the YouTube works here or not, but I suggest all of you to take a YouTube. Maybe I don't know how, Nidhi, how am I doing with the time?
So we have the time. Yeah, we will good.
Okay. So I would suggest I would suggest all of you to go through he's not going to play here, and this presentation will be made available to all of you. So in this case, what happens is, for example, this is what I'm showing for a cartilage and knee replacement. So from a knee, samples are taken, biopsy is taken from a patient, and the cells are expanded in a culture dish. The cells are isolated, and the desired cell type, like chondrocytes that I mentioned, they are cultured, expanded, and a synthetic knee capsule is created using matrices, different matrices and different cell types together. A organ-like structure is created, and that organ-like structure is cultured in a culture dish in a big well, and they are made to mature. The maturation of this organ is in such a way that it closely resembles what in vivo in the knee capsule that has happened, that is happening. So once this organ is taken out, once this organ is developed in vitro, this is taken up and implanted in this kind of technology. It is much more complex than cell-based therapies. It is very early on. A university called Wake Forest University in North Carolina is doing some of these studies. There are some, they are able to create structures like artificial lung. They are able to create urinary bladder. They are able to create mammary gland, right? So these are all in the very, very early phases. There is a sufficient investment into these areas in cell therapy. At some point of time, if you are able to make this kind of organ-like structures, and you can get them back. And the other area that happens in this kind of tissue culture, most of you students may know what is a two-dimensional culture. That is, you use cell suspension, put in the culture dish, and then in the media, in a regulated environment, and these cells grow. The cells grow as suspension cells like hematopoietic cells, or adhered cells like the mesenchymal stem cells. In this case, instead of that as a layer, they put as like a spheres, like a organ-like structures. These are called 3D cultures, organ type of 3D cultures. That is also evolving very heavily in this kind of tissue engineering area that is also on the progress on that. So I come back to the nitty-gritty details of, you have this knowledge, you have trained in MSc, you are trained as a BS, a Bachelor of Science or Master of Science in a specific area of biochemistry, life science, or whatever is that. So what happens when you come to an industry, right? So you are looking for an opportunity, how to take these cell therapies on that, right? So if you look into just having the fundamental degree is not alone enough in this area of emerging areas. Particularly the syllabuses and the outline of courses that one has now have to be completely updated. Some universities are happening. A lot of universities are not happening, as I see it from a different atmosphere from India to here, right? So, but if you take these ones, if one has to enter into an industry like cell therapy or regenerative medicine, there are several things. You need a lot of competences. What are those competences? It is not enough if you learn and get a gold medal in your class and all that. You need really interpersonal skills, meaning that you should be able to work in a team, right, very nicely, and integrity. So you should be very honest with all this. And you need a really professionalism in presenting it or discussing it or presenting it. Instead of personal questions, you should ask why this is happening, instead of who did it wrong, right? So you need to have initiation, dependability and reliability, and lifelong learning. So this is the most important feature in my opinion, not just for the course of a degree or not just for the course of a syllabus. You need to be learning virtually every day on this kind of things. So you have competence, academic, this is most important compared to any other kind of competencies that one needs to work in the industry or in academia and all that. So you know the academic competencies. I'm not going to go through academic competencies. You all have passed that stage. And workplace competencies are this thing. In order to not only work in terms of science, you need a good teamwork. You need to be planning and organizing. You need to be innovative thinking. What comes next should be the question that you should be asking. And then problem posing and problem solving. These are the kind of things that you need. And you need ideation. So you have a problem. Yeah, how do you solve it, right? So those are some of the things that you need to have it. Working with different tools and technologies. None of us were efficient on Microsoft Teams or Zoom or any kind of these technologies without the need of without the force of COVID-19. So now everybody is proficient in this kind of emerging technologies like Zoom presentations and all that. Checking and examining and recording. Business fundamentals. Yeah, in a kind of business environment, you have to work in a kind of industry. How do you work? What are the business fundamentals that you need? So you need real technical competencies like fundamentals of bioscience, research and development. You would see in the next slide on all these kind of things. Manufacturing ability, quality control, health and safety. These are all the different areas of expertise that you need if someone has to launch a product, if someone has to release a product in the area of cell and gene therapies, regenerative medicine technologies. These are not only this technology, the gene editing technologies, and also they are expanded into agriculture, expanded drugs and pharmaceuticals, and even into some kind of medical devices and equipment. These are all industry-related technical device-based competencies that you need. So after all this pyramid, you need the management competencies and occupation-specific requirements for all this. So these all is needed. How do you do it? So you need really good foundational workplace to learn. And if you are looking for a job, you need job-specific tool talent that you need to develop either by working as an intern in that industry that you plan to go. And then you have to do that in a safety and skilled, you have to accumulate a kind of talent and skill set that could be used for a ready to use in the workspace. So mainly, this is mainly like safe, skilled, and ready workforce program that you need. And this is here I will bring up why I was attracted by Professor Redena's initiative on FABA Academy is creating this kind of environment to creating a liaison with industry and identifying all the industry within India or abroad, and then bringing students, giving a kind of a window shopping look on what they are doing, whether they would fit their thing and match those interests and create those kind of environment for training and possibilities on those. So if you take this, for example, in a typical biopharma workforce, I've taken as an example for typical biopharma. So the activities they do in a regenerative medicine based company is manufacturing for clinical use, manufacturing commercial in a kind of allogeneic therapy, manufacturing for other, and there is an R&D that is associated. This is the effort that you see. So what it means is, let's say in R&D, you go in R&D, they only need 8% of these activities. 60% majority of the activities in the manufacturing, right? On the science and technology side, if you take it in particular, if you take on the technology side and the functional areas, the manufacturing is still the big base. And then facilities and engineering, you need to create a GMP facility. You need operation force who can manufacture. You need a quality assurance. Now you have made a product for, let's say, critical limb ischemia, whether this product works for the indication, you need a QC and quality control. And you need, after you make all this, you need a commercial sales force to go out and sell this product to make money for the company. And education and employment, if you look into the education and employment, so this is, the MScs and PhDs. PhDs are less force in terms of industry. They are needed, but they are less force. MScs and Bachelor of Arts, all technical jobs are taken up by Bachelor of Sciences with training. Similarly, validation in QC and QA, people take all Bachelor of Sciences for this. So, for everybody, there is a role. For everyone, there is a role that if you want to, right? So set a mind onto it and approach people how you can develop those skills that you want to go in terms of these areas, and keep track of what is happening in these kind of areas. So with that, I come to a conclusion. I think the regenerative medicine technologies mainly consists of cell therapy, gene therapy, and tissue engineering. And gene therapy involves the technologies such as gene engineering using CRISPR-Cas9, TALENs, or zinc finger nucleases. For future workforce in regenerative medical technologies, apart from the academic degrees, you need to have hands-on training skills to be able to get a job in these areas, or to start your own industry, like what Manohar did. You can start your own industry for a specific area. You need expertise in those kind of specific areas, in addition to academic credentials. I think I would stop at that point and take any questions, maybe.
Thank you, Dr. Mohan. It was a wonderful presentation.